Very good. It is 3 minutes past the hour, so maybe we should get going, and we'll probably have some guests signing in and signing off throughout the seminar. So yeah, first today we're going to have some opening remarks from Dr. Hui-Ching Lee, who is the project scientist from NIH assigned to the INCLUDE Data Coordinating Center, and she's with the National Heart, Lung, and Blood Institute. So the podium is yours, Hui-Ching, for any opening remarks.
Thank you so much, Joaquin. Good afternoon, everyone. We're so happy to see some of you in person last week. So let's share my screen. I will give you a very high-level overview of the new features from the Data Hub and some new activities coming soon in the near future. As you may know, the mission of the INCLUDE Data Coordinating Center is to accelerate discoveries that continuously improve and enrich the lives of people with Down syndrome through accessible data resources that enable interdisciplinary collaboration, connected communities, and the expansion of a global Down syndrome knowledge ecosystem. So today I want to share with you a few highlights of the new features of the Data Hub and a few features coming soon, in addition to some opportunities through the NIH INCLUDE project. The first new feature is that on the DCC website, you may notice that we now have a brief summary of studies, not only that are for studies that are already in the Data Hub, but also some upcoming studies in the Data Hub. Another new feature is that the aggregated data and the study pages are now more visible without logging into the Hub. So we have register data and we have control access data, and those the landing page now currently have some information for you to check even without logging into the Data Hub. And I want to mention that currently only harmonized data is available, and in the near future non-harmonized data will also be available soon. New features for the portal cover the cohort builder, the virtual biorepository, the study page, and the variant explorer, and will be presented by the DCC team later. As Dr. Bianchi mentioned on the INCLUDE investigators meeting last week, we will have the experimental models of Down syndrome research portal coming soon. Key features include a study finder, exploration portal for animal models by gene, protein, and metabolite, and also by tissue, organ, and cell type. Another major activity is the implementation of NDA GUID, the National Institutes of Mental Health Data Archive Global Unique Identifiers, so that researchers can use linkage strategy to remove duplicates in a synthetic cohort using data coming from different Down syndrome cohorts on the Data Hub. The GUID map is controlled access and can be requested through dbGaP in the near future. The INCLUDE DCC is now fully implemented the Research Authorization Authentication Service called RAS, and the controlled access data can be distributed through Kavatica soon. The RAS implementation also makes the Data Hub interoperable with other major NIH data repositories. Last but not the least, I want to remind you to check the INCLUDE funding opportunity page for your grant application. I want to remind you that we still have 2 receipt dates for the XO1 NOFO and in the next 2 years. And the R3 has a receipt date this year on July 2nd. And the R3 NOFO will be renewed. So don't worry if you see, okay, I didn't see a new R3, we'll have that in next year. And the final reminder is the Include Cloud Credit Pilot Program, which is still running, and we welcome cloud credit request. So I want to say that we are so glad to see that I got two requests for cloud computing after last week's Include investigators meeting. All right, that's all I have. Thank you.
Thank you, Hui-Ching. And if anybody has any questions about any of the presentations, you can enter those here in the chat, and we'll be monitoring that. My name is Joaquin Espinosa, and I'm the leader of the Administrative and Outreach Core of the Include Data Coordinating Center. And I have a few slides to share with you before we get into the fun part, the more juicy part of today's webinar. So welcome to the Spring 2024 webinar of the Include Data Coordinating Center. And this will be a bit of an interactive presentation. Throughout my slides, I'll be asking you to take your phones out and scan some QR codes, and Haley will also be posting some URLs for you all there in the chat. As Hui-Ching mentioned, we had the first ever in-person Include Investigator meeting last week. Many of you may have been there. It was a great meeting to celebrate the 5-year anniversary of the Include project. We have diverse presentations covering the full spectrum from basic science to clinical trials, career development, and so on. Very strong representation of the projects funded by the wonderful Include project, including many international collaborators, and I see many of them already here in this presentation today. Overall, it was a great time. So we were very happy to meet many of you in person there. So here's the first call to action for you. I want to promote the Include DCC website. Of course, the Include Data Hub is where all the data is and where you can make great discoveries in service of people with Down syndrome. But I want to make sure that you know we have the website where we share with you a lot of information. This is includedcc.org. And something that you can do to make sure that you are updated of any developments, any new data sets, any new webinars, is to subscribe to our newsletter. So you will find that in the website. It's very easy, it's on the front page. Now, you may or may not know that every Include funded investigator, every PI, has a profile in the website. For example, I'm sharing here some screenshots of Philip Lupo's investigator profile. You can see his grants, his publications. Now, we need your help reviewing this because this is populated in a semi-automated fashion. We want to make sure that you like how you're being profiled, that you like the grants. If we are missing any, you can let us know. If there are some Include funded publications that we haven't captured through our method, go ahead and let us know. So please take a look at your profile. And if you have any questions or modifications, you can email us at info@includedcc.org. I want to brag a little bit about the powerful multidimensional data set that you can find now in the Include Data Hub. This is a first-in-kind data set covering 11 different studies, more than 9,000 research participants, more than 7,400 of them with trisomy 21. And I'm listing here the logos and the number of participants from the various studies, and no study is too small. We have studies with thousands of participants. We have some with dozens of participants. We don't care. We love them all. We want to make sure that they're all represented. And I'd love to update this slide with more logos, more studies as we have more data generators contributing their data sets to the hub. And also it's a very multidimensional data set. This is very unique when it comes to data resources out there. We have the clinical profiles. Now we have passed the 4,000 mark for whole genome sequences, more than 1,700 transcriptomes, hundreds of protein metabolome. And if you look at our pipelines, we have many other data sets. So we hope that you will continue to come into the Data Hub and benefit from these data sets and become secondary data users of these Include funded research. So here's one of those QR codes. If you have your phone, you can scan this and it would take you to a form that you can fill up. we are looking for Data Hub users who like to share their experiences with us. We build it, you've come, now we need to make it better. We need to make sure that the Data Hub addresses your needs, that it has the features that you want, that if there are parts of the user interface that are not friendly to you, that we make them more friendly, what have you. So I want to personally acknowledge, and express my gratitude for these users, Data Hub users that are pictured here at the bottom of the slide, because their feedback has been invaluable. Every one of these individuals has some really good ideas about the user interface, the data sets, how we build the Data Hub, and we are surely incorporating that feedback into the hub to make it better. Now, if you are not yet a super user of the Data Hub and you are struggling, finding things or how to make it work for you, let me remind you, we have office hours with a team of experts every third Thursday of the month, 2 to 3 p.m. Eastern time. The next one is May 16, where you can join, and you'll be a one-to-one workshop. You're going to a breakout room, one-to-one interaction with an expert that can help you find the data that you're looking for, push it to a cloud workspace, or whatever you may need. So please mark your calendars. If you sign up for our newsletters, you'll be reminded of these monthly office hours. I also want to share that we'll be at a lot of conferences this year, and that we would love to connect in person with you. So we are here. We just concluded the investigator meeting. We'll be in Rome at the Trisomy 21 Research Society meeting. So we hope to see many of you there. We're going to have monitors to demo the portal. We're going to have some presentations about the Include DCC. So looking forward to meeting with you in person. And then these are all the meetings that we have planned here for the rest of the year. So if you happen to be attending any of these, again, looking forward to meeting with you in person. Also, for those of you who have trainees that may benefit from greater data science literacy, the Include Project funds this Data Science for Diverse Scholars in Down Syndrome Research course. This is a 2-week immersive hands-on course during the summer at the University of Colorado campus, University of Colorado Boulder campus. And, we provide training in data sciences to graduate students, postdoctoral fellows, and early career investigators with a focus on those for underrepresented minorities. We also provide some training in career development skills. And one of the key goals here, of course, is to make sure that we have a very diverse next generation of Down syndrome researchers that can bring new ideas and new approaches to the study of Down syndrome. And I want to again express my personal gratitude to all the guest speakers for this upcoming version 2024. They're listed here, a very diverse group of speakers presenting on different topics. So we already found our 25 students, trainees for this year. So we're done for this year, but please let us know if you want to be thinking about sending somebody for 2025. You can scan this QR code. Haley will be sharing the URL. We now have multi-year funding to be able to provide this course every year moving forward. So if you missed it this year, there's an opportunity to send your trainees next year. And then finally, I think this is my last slide. Again, sign up to receive updates on our upcoming events, any new data releases, and any new relevant activities. And that is my last slide. I'm going to stop sharing. Now it's my pleasure to introduce Dr. Adam Resnick, who is the leader of the so-called Data Portal Core of the Include Data Coordinating Center, and he'll be sharing with you all some cool new features that we have brought into the portal in the last few months. Adam, the podium is yours.
Thanks so much, Joaquin. And again, let me echo both Wei-Ching and Joaquin's sentiments that it was really fantastic to meet many of you who are likely on the call in person. last week. And already, I think our teams are ideating and acting upon some of the suggestions and opportunities that were enriched through our interactions. As part of that meeting, we had the privilege of giving you a brief 5-minute intro across the Data Hub. And today, what we thought we would do, at least from the portal perspective, is expand on that and do an actual demo of the portal features that are newly enacted and available within the portal in ways that hopefully set the stage for you to better use the portal, particularly as we continue to release studies and or set the stage for you to come join one of those office hours that Joaquin mentioned. So as Wei-Ching mentioned, there are 4 different exploratory tools that we're continuously innovating and deploying, and I'll highlight those features as part of this. And this includes, the cohort building activity of the portal that allows you to build cohorts based on biosamples, participant features, and file types. A virtual biorepository feature that allows you to actually interact with and potentially create a cohort for submission to participating biorepository. A new feature that allows us to represent features about a study in a comprehensive and unified way. And then the Variant Explorer, which is a framework that allows you to essentially review variants across studies that have molecular features of that nature and explore the occurrence of those features. So I'll start with a couple of slides and then go to the portal. So the cohort builder really functionality has this context that allows you to combine features across different types of data or elements within a study or across studies. So I'll go through this, but the key framework is that within any one context, you can an and or an or a set of features, or across multiple contexts, combine queries and save them and reuse them. And then this is complemented by a set of features that are displayed in graphical format in ways that also allows you to further filter these contexts. And so that's one of the key frameworks of either selecting or creating cohorts. The other builds on the extensive harmonization that we do across the phenotypic landscape of studies for those studies that we can harmonize such features for. And in that context, there are these 2 additional browsing features that allow you to explore based on one of 2 ontologies. These ontologies allow us to contextualize particular features depending on the level of specificity provided by the study team in ways that provides you another filtering activity for the cohort, and I'll demonstrate these as well. So it's this combination of phenotypic and diagnostic feature selection, plus a combination of essentially and/oring combination of features across multiple studies, participants, or files that allows you to create a cohort that you can then interpret in terms of the availability of participants, biosamples, and data files. So let me switch to the portal and just show you a few views from that. So the starting point for many folks is really at the includedcc.org website, which Joaquin highlighted to some extent. And from here, you can just click on the Data Hub portal. Let me actually log out so that you guys can see the initial view. And as Wei-Ching mentioned, you can see some of the features and representation of participants already here. So these numbers will change across times as more and more cohorts are onboarded, as well as some metrics associated with them. And from here directly, you can launch into the Variant Explorer and the Cavatica workspace, which I'll touch on briefly. But in this context, you can log in with either your Google or Login RD. And then once you log in, you'll have your own save state environment that will bring you to the dashboard context. For cohort building, you can typically begin with the Data Exploration tab, which is on the top here. And within that context, the first place many people begin is really the colorful graphs that are available here. And again, these are both to highlight the diversity and feature set of the cohorts, whether that is at the phenotypic or diagnostic context, or some of the demographic or file-specific features here. And all of these charts also act as potential feature selection as part of the cohort building. So for example, if I hover over any one of these features, you'll see how many participants fit within the feature. So I can, for example, select congenital anomalies. It'll select both the chart, but I can also add that term into the query above, and you can see there are 3,600 or so participants with this Mondo term available. This is not the only way to select those features, and I'll highlight that in the context of the faceted search that opens up when you click on each one of these features. So let me create a new query. So here, through the faceted search, you can query along any one of these dimensions as sets of features. And before I go here, let me just highlight a feature here. So I initially selected a developmental phenotype of congenital anomalies via the Mondo term. I can also specify that I'm only interested in participants that are annotated as trisomy 21. And so you can see I removed the 13 participants that also have congenital anomalies but are not trisomy from that cohort, right? So I'm ending across that context. But as I mentioned, a second interface that provides you greater granularity across the phenotypic and diagnostic landscape is exploring it through either one of these two faceted searches. So in this context here, you can essentially just begin typing or explore the ontologies. And this is where the power of harmonized ontologies really comes to fruition, because it allows us to essentially pick terms that map to the study level of specificity. So for example, a study may designate a feature as nervous system disorders, while another study may actually provide a more specific type of nervous system disorder in that context. So depending on the level of specificity applied at the study level, we can bring that view into this context. And here I just started with celiac disease, as a feature, as a common. And a couple of things you'll notice is that there are two columns here. The left-hand column really designates the number of participants for which the studies specifically, annotated the phenotype or diagnostic feature on the left. The right-hand column really is a summation that accounts for those features as summed up to the highest level terms. So for example, disease or disorder, there are 7,400 participants that match that upper level term that then gets specified across the studies as you filter them down. But you can essentially bring as many studies into this context. You can and/or them as well within this view, and then apply those terminologies to the features here. And here you can see I applied it within the context of that same query at the top, right? So I essentially added features through either the graphical interface, the diagnostic or phenotypic exploration context, or the faceted search directly. And these can be appended together to create that framework. And you can continuously actually modify those as well. So you can also move to a second query. So oftentimes, as you're beginning to think about queries, you'll combine feature sets that make sense to combine, and then you'll start a second query in that setting and begin an alternative context from both a phenotypic or demographic perspective. And again, it's very similar in context, whether you're using an HPO browser or the Mondo browser as a context. And it just depends on the study level metadata that is being applied in that setting. But once you create a set of queries, whether it's that demographic or, as I'll show, a biospecimen or data, you can also combine these features as a context. And in combining them, you can then and/or them. One thing that I do is I actually name my filters in ways that I can come back to them. So once you have a set of filters, you can always come back to that filter, and these numbers will constantly update over time. So if you have your favorite sets of phenotypes or diagnostic phenotypes that you care about, or any features, this saved filter set will allow you to always come back to those views in ways that is very facile in nature. Any cohort that you create can be further explored at the row level data for those participants by clicking on the participant tab here. And this is where you'll actually get participant by participant features defined by the cohort building activity. And you can get greater granularity by clicking on the participant themselves in ways that will provide levels of information about that participant beyond just the cohort features you define. So here you can see that there are additional Mondo and HPO terminologies with this particular participant and their association. And different studies will have different components of the profile, summary, diagnosis, phenotype, biospecimen, and data files collected depending on the study composition. And you can also directly download a selected participant and or their family members as a file for use in other contexts. So let me create a new query and go to the setting of, let me modify actually this query just to show you the modification process, and go to the biosample context feature sets. So in this setting, I can actually explore the biosample features directly from the faceted search. So here I can either search by specific sample IDs, collection IDs, or saved by specimen sets. Most people do not have the saved sample IDs or collection IDs until they get further along in their studies and are working with a particular cohort, and in which they potentially want to monitor or track if an additional sample might have been used in another study for another data generation project that they may want to integrate in the future. So this ongoing scientific process of leveraging samples and tracking their use across studies is one feature that is built into the portal context. And this is especially aligned with the idea that we want to essentially support a virtual biorepository network, which we've already begun piloting in this context. So within this setting here, when you go into the participants, you'll see a listing of participants. And I'm just going to go ahead and select some of these participants. This is another selection feature that allows you to essentially work with the participant context. You can save a participant sent, just like I saved the features previously, or you can add it and remove it from set. So you can essentially continuously modify and in a granular way, for example, remove a specific participant that doesn't fit criteria for a particular reason from a cohort building activity that you have here in this context. So for example, I can remove these two participants if I now think that they do not meet a certain criteria from a particular set that I've saved previously. Importantly, the associated biosamples for the participants are also annotated. And this includes both the biosamples from which data has been generated in this context, which oftentimes these are the aliquots that were used to generate the whole genome sequence or proteomic data files, but also the categorization of aliquots and biospecimens into studies in ways that permits you to see whether or not a particular plasma collection and the peripheral whole blood component from that initial collection still has available aliquots, despite the fact that there are already two files associated with it. So here, as an example, you have a sample ID that annotates an aliquot of plasma from a peripheral whole blood collection event that has still samples available in ways that we can walk through in a second for a particular request. You can also directly see which files are associated with that plasma analysis. And in this context, it's a metabolomics and proteomic context as a setting. But within the biospecimen setting, you can essentially select, for example, in this context, the plasma aliquots that are still available, and actually click on a request biospecimen for those studies that have a virtual biorepository context. And in doing so, you will be able to save it for future use and working. And that we recommend that you keep track of this, in part because many biorepositories will refer to this manifest as the source of interacting with the user who is coming to that repository. So here I'm going to open up the request so you can see what's included in there. So one, there'll be a README file that provides you some information about the composition of samples. In this context, it's the Human Trisomy Project under CERNIC, and I'll give you a little bit of information about where you want to go to request those samples and where you'll be able to essentially take the manifest, which is the second file that's available, and upload this manifest into the repository request. So again, here's the contact information, recapitulating some of what's in the text file, but here is that biosample request that you can essentially append and attach in the context of this repository to a request process that then will be mediated by the CERNIC team and iterated on. So this is the second context I wanted to highlight that we discussed as part of the biorepository workflow once you move through the cohort building process. So let me move into the next feature, which we highlighted as part of the meeting, which is really the Variant Explorer. So a large number of studies that will be coming through and already are part of the INCLUDE program contain molecular features of a variety of different data modalities and types, as Joaquin mentioned. One of the challenges that we are trying to solve for is as you begin to assemble multiple studies that are individually governed by different dB-gap accession numbers and processes, how can a user who might be genomically inclined figure out which studies they should apply for dB-gap access, which combination of studies they should combine if they're interested in a particular feature of data from that study. The Cohort Explorer context provides you a framework of creating features at the biosample layer across studies or at the phenotypic layer across participants. But we wanted to solve for the challenge of creating cohorts and understanding which cohorts you might want to create in the absence of a user yet having direct dB-gap access in ways that would allow them to run genomic analyses. And this is what the Variant Explorer context provides. All the data that you see available within the Variant Explorer actually is derived from the files that are available as part of the studies within the context of genomic data. So in the context of the genomic data that's currently available here, for example, these provide you access to the source files themselves, for example, the CRAMs, the GVCFs or VCF files, but the extracted feature set of variants is what flows into the Variant Explorer for those users who want to begin exploring the variants themselves before interacting with the specific source files. You can see here, I do not yet have authorized access to these files. I don't yet have the dB-gap permissioning linked to these. But in order to understand what's available within these data from a variant perspective, I can explore that data through these filters here. As you can see, there's a very large number of variants to explore, more than 74 million just across the 11 studies. And these include all variants in this context, not just those that are annotated as pathogenic, but that's really the context that most users begin with when they explore this setting. They begin from two lenses to understand what might be of interest. The first is to ask the question around things like, of these 74 million variants, how many of them are already known to be pathogenic? And are there any variants of interest in that context? This provides really an updated view of ClinVar and some of these pre-computed annotations that users can filter against. So for example, I can filter across all pathogenic or likely pathogenic variants across the data sets, and you can see there are, within this context, there are 1,500 variants across a number of genes, across a number of studies in this context. And again, the same process is invoked in this context, where each of these faceted search allows you both a way to further filter the data, as well as apply additional filters in a sequential manner. So here in this context, I selected pathogenic or likely pathogenic. I can create a new query and leverage pre-computed features that many genomic researchers utilize. For example, I can employ a CAD score, which is really a summation of a variety of different pre-computed features that interpret potential harm of a variant. So I usually use greater than 25 to be selective as a context. That still provides you a large number of participants, even with that CAD score. Likewise, you can essentially include additional features from the variant perspective in terms of consequences of those variants and filter by those settings. So for example, you might only be interested in missense mutation or splice region mutations or stop gains, as a context. And again, you can filter to those features. Then likewise, you can also include features, in terms of specific frequencies across either NOMAD, TOPMed, or 1,000 genomes as a framework for additional selection of those features moving forward. Like in all the other contexts, you can continue to either add or combine these features in ways that can continue to essentially filter the features. So here I combined my query for pathogenic, likely pathogenic, with specific missense, splice region, or stop gain features as a context. And when you do that, you ultimately begin to hone in on a specific variance of import to you. And once again, you can save this filter set. Like in all the other feature settings in the other tabs, you can also, again, further refine at the variant level or interrogate a particular variant and its annotation framework, its occurrences, where this variant is occurring and in what context, from which studies, in ways that you can have deeper insight into it, and once again, make a decision as to whether or not you might want to apply to the DSCOG-ALL study as a cohort. And this, by clicking the DSCOG-ALL, you can again see all the data, participants, biosamples, or data files associated with that study. So within each one of these views, you can click on any feature to add it essentially into the queryable context, and then further refine it by, again, doing combinatorial data. And you can see that combining these two sets of celiac disease, congenital anomaly with the ALL cohort did not produce any features specifically as a context. Because there are these multiple views, again, we recommend that you save your filters and across both the variant context as well as the data exploration context in ways that I think can be very powerful for ongoing research as more and more studies come into this context. Let me highlight the next feature that we recently implemented that Wei-Ching also described, which is the study page that which I think I have up here. Oops, I missed it. That allows you to essentially really review a comprehensive setting for the studies themselves. So many users actually think about studies from a study perspective. And right now, as Wei-Ching mentioned, all of the studies essentially have this harmonized layer where we've harmonized the phenotypic data, genomic data, molecular data set that allows these cross-cohort building. But we recognize that that won't always be possible, and that in many ways, we want to release data in a much more accelerated fashion, even as we work through that harmonization. So very soon, you'll see additional rows here of studies that have not yet to be harmonized, but the submitted data will be available for you to explore. And you can explore that data essentially by clicking on a particular study and then seeing in quite detail what's available within that study in ways that you can both filter across that context and select features, or further interpret the context of the study and what's available from both a data, participant, or file scenario as you're working through the availability. So as both Wei-Ching and Joaquin mentioned, we are constantly revising and informing the portal, and we'll be deploying additional features in an ongoing fashion. Joaquin provided you a sign-up page to provide direct feedback for the portal. But one of the things that I still do in addition to that is use this feedback tab on the left-hand side. So this feedback tab allows you to essentially capture any view that you're in and provide feedback to the portal team, and in a way that can connect you to the portal team with a very granular view of what is the part that you have a question about or need help with, adding that screenshot to the context and submitting that to the portal team. So that's just another avenue of both informing and potentially connecting in a very refined fashion around a specific feature of the portal. But please also sign up for that feedback across the data hub writ large through what the link that Joaquin provided. And so with that, I'd like to thank what is really an amazing partnership across several entities, including us here at CHOP, the St. Justine Hospital, where Vincent really is the champion and leader of the portal development framework, and Valsera, who essentially supports the deployment and feature integration of the compute environment in Kavatica, which we did not go through today, but will in future context. Thank you so much for the opportunity to present.
Thank you, Adam. I always learn something new about the Data Hub when I listen to you do these demos. And we're doing well on a schedule. Let me remind you that we have 2 scientific presentations from INCLUDE funded researchers who have data in the Data Hub. But we do have time. If there are any questions for this first part of the webinar, please feel free to use the chat function here. You may also raise your hand, and I may be able to detect that you've done so. Any questions for Adam, for Wei-Ching?
I realized actually just now that in my excitement to show the new study page, I skipped over the second entry point. We oftentimes see users interact with the Variant Explorer, and maybe I just want to highlight that. So, one view of the Variant Explorer that many users begin with is, as I mentioned, let me click on the right tab, filtering across features that allow you to identify specific threshold of consequence in ways that can prioritize variance in this setting. But oftentimes, I think many of us are also gene-centric in our approach. So here again, you can potentially choose your favorite gene of interest, as a context. Like I just chose GSChem, and this will tell you that there are 28,000 or so variants for GSChem. And here you can also add additional filtration components from Nomad for loss of function constraints. So for example, you can, for the loss of function context here, I think most people use for the, I usually use 0.35, as a context for this one. And I think most people like to start with greater than 0.9 of the feature set. So you can add also sort of loss of function constraints around specific genes, as a context for filtration, from a gene-centric view in this context. And obviously, again, you can further refine with additional scoring or ClinVar variant applications that are applicable to these 21,000 variants in this setting. So I didn't want to miss that, because I think some of us come from a filtration by variant, some of us come from a filtration by gene-centric view.
Thank you, Adam, for that. Yeah, I'm a gene-centric person. I'm on a mission to memorize all the acronyms for all the genes in the human genome. I need another 40 years, but I'm good. Very good. Okay, without further ado, then we'll move on to the scientific presentations of today's webinar. And the first one is from Dr. Kelly Sullivan, who is an associate professor in the Department of Pediatrics at the University of Colorado School of Medicine. And want to thank Kelly for willing to present unpublished results at this webinar. We welcome that very much. And I think Kelly will do a great job at illustrating, how to use data in the hub, but also the value of some of the upcoming activities, such as the experimental models portal. And Kelly will be presenting on cross-species analysis of liver dysfunction in Down syndrome. Kelly, the podium is all yours, and then we'll have time for questions after your presentation.
Wonderful. Can you hear me all right?
Yes, loud and clear.
Okay, great. Well, yeah, it was wonderful to meet so many people last week in Bethesda. I think that was a wonderful meeting, and I look forward to hopefully many more of those in the future. And I'd like to thank the DCC for the invitation to join you all today and share a little bit of a story that we've been developing in my lab at the Cernic Institute. Great. So I just want to start at the beginning and kind of move through, how we're thinking about things in my group and trying to understand Down syndrome. So I think we all know that trisomy 21 or 3 copies of chromosome 21 is the molecular cause of Down syndrome. What I think is less clear sometimes, particularly to newcomers in the field, is that we still really don't have a good handle on how we get from the 3 copies of chromosome 21 to Down syndrome. We have this kind of black box in the middle here. I think part of this is understanding what it is that makes up Down syndrome, right? Which is this unique disease spectrum. So this increased prevalence of CHD or severe respiratory infections, Alzheimer's disease, autoimmune conditions, leukemia, many more conditions that are more prevalent in the population with Down syndrome. And then on the flip side, these decreased prevalence of things like coronary artery disease and solid tissue tumors. And so, the question we're really trying to understand is how might an extra copy of chromosome 21 be driving this increased prevalence? And by understanding that, maybe we can understand how to ameliorate some of those conditions and to improve and expand the lives of individuals with Down syndrome. So another way to look at this is kind of from an organ-centric view. And the only conclusion one can really draw from that is that trisomy 21 adversely affects basically every tissue and organ system in the body. So I think we're all familiar with the neurodevelopmental and neurological impacts of trisomy 21. We also have cardiovascular anomalies, skeletal, hematological, craniofacial. Basically everywhere that we look, we're seeing that the trisomy is having disparate effects on development and pathophysiology in Down syndrome. And so what I'm really interested in understanding is how can we study the complex cause-effect relationships between trisomy 21 and the pathophysiology that we see in these various systems. And it can be really difficult to obtain primary samples from the vast array of different tissue types that are affected by trisomy 21. And so what we need to understand this is a series, I think, of complementary experimental systems that includes primary human samples, human iPSCs, and animal models of Down syndrome, for example, as just a few prongs that we can use to kind of uncover what's in that black box and understand more about what's going on. So each of these systems have pros and cons, but together we think they're a really powerful discovery engine. The integration of data from different sources and experimental models can accelerate discovery and identify putative mechanisms of pathophysiology. Oh, we still have people trying to get in from the waiting room, and that is preventing me from advancing my slides. Okay, back to this. Okay, so I'm going to tell you a little bit about a story again that we've been developing that I think integrates, hopefully effectively, data from these 3 different modalities, where we're looking at altered hepatic signaling and liver function in Down syndrome. So let's start at the beginning, and identification of putative altered hepatic signaling in Down syndrome using primary human samples. And so I'm at the Cernic Institute, and so our favorite, starting point for human data is the Human Trisome Project. So just to orient everyone to what that is, this is a massive panomics cohort study with what we call deep clinical metadata, which means when our individuals come in and join the study, we ask tons and tons of questions about the different co-occurring conditions, so that when we get our blood draw and we generate whole blood RNA and plasma and blood cells to do our omics exercises, we can actually generate biosignatures that are associated in a kind of a one-to-one manner with the different conditions from each of those individuals, right? And so we really seek to integrate these biosignatures that we're generating from the omic data with the clinical reality from these individuals in hopes that we'll identify some interesting patterns and understand a bit about what might be driving these. So again, we can understand what potential pathophysiological mechanisms are at play, but also how we might be able to ameliorate some of those things. And I just wanted to mention, again, we are big fans of open data. So all of the data that I'm going to show today, at least on the human side, are available in our own portal, the Trisome Explorer, which is available at this website. And then, of course, we've deposited everything into the Data Hub. And so all the data, again, from the human side that I'm sharing today are queryable through there. And so just to come back to this idea of different tissue systems and how they're functioning, and understanding, again, from these primary data sets where we can glean the most information. Again, the whole HTP, all these blood samples and omics from our group and from others are invaluable sources for kind of hypothesis generation. But I'm really starting to fall in love with the information that we can derive from plasma in particular. And the reason for this is that, plasma is kind of a collecting duct for all kinds of different things, right? So we have information entering the plasma from the brain, heart, lung, liver, kidney, kind of you name it. And some of these are secreted proteins that we know are signaling molecules, growth factors, those kinds of things that should be made, generated, secreted into the blood. The other thing we actually capture in the plasma are signs of damage, right? So when we have damage in these different tissues, when the cells are dying and things are being released, those end up in the bloodstream and they get captured in the plasma. And so there's a really powerful matrix of information that we can use to understand how the different tissues are functioning. And again, we can do this without having to biopsy these individuals. We take the one blood draw and we capture information about all these different tissue systems. And so this allows us to kind of build out some of our observations. So to start with, we performed untargeted mass spec plasma proteomics. And so we just took the plasma from these individuals and we measured a bunch of metabolites, and we asked kind of what are the global patterns of all individuals with Down syndrome in this cohort, about 300 of those versus around 100 control individuals. So here we're just starting with a waterfall plot where we classified these and said, what is the overall behavior of some of these classes of metabolites? And what we see here at the top is a pretty strong dyslipidemia. And then one of the things that jumped out right at the top that we were very interested in is bile acids. And here you can see that all the bile acids we measured are actually upregulated in the plasma of individuals with Down syndrome. This is actually the most significantly abundant metabolite in our analysis, taurolithicolic acid, which is one of these bile acids. And you can see here that it's substantially elevated in individuals with Down syndrome. So why did this catch our eye? Well, bile acids are a really integral component of liver function. So these are solely synthesized in the bile by the hepatocytes, and then they are actually sent to the gut where they do most of their work. They're involved in the digestion of fats and intestinal absorption of other hydrophobic compounds. And then they enter hepatic circulation. They kind of circulate back and forth, between the gut and the liver, where they also serve then to regulate liver function, basically acting as hormones and signaling through membranes and nuclear receptors. And so this was our first real hint that there might be something going on. And as we started to consume the literature, we realized that elevated plasma bile acids are actually a pretty strong hallmark of liver dysfunction. And this could be associated, again, with some aspects of that dyslipidemia, right? One might imagine if we're not metabolizing and absorbing our fatty acids in the right way, we might see some alterations in the abundance of those when we look in the plasma. Great. So we next looked at the plasma proteomic profile, and we just started by asking, about some known markers. So we're looking here at ALT and AST. These are enzymes that are expressed in the liver, and these are kind of canonical markers of liver disease and liver dysfunction. And you can see here that neither of these are elevated. This is consistent with reports from another number of other groups. But when we looked at some other markers of liver function, we started to see signs that things might not be quite right in the population with Down syndrome. So here we're looking at albumin, which is just a carrier protein that's synthesized in your hepatocytes again, and then secreted into the blood where it serves a carrier for a number of different types of molecules. And you can see that we have lower levels of albumin, which is, again, a hallmark of liver dysfunction. Then also lower levels of insulin like growth factor 1, again, which is a growth factor that's made about 75–80% made in your hepatocytes and then secreted and does a number of different functions throughout development of different systems in the body. And so, just from a kind of a biased viewpoint here, when we asked about a number of factors, we had kind of 50-50, a couple of canonical markers that are not changed, and then a couple of other markers that are changed. So we next took an unbiased approach, and using ingenuity pathway analysis, we just put the whole list in and said, okay, which are the pathways that are perturbed? And what we found was actually quite a lot of things that are directly liver associated or at least liver related at the top of this. So LXR, which is the liver X receptor, or RXR activation, coagulation, hepatic fibrosis and stellate cells. Stellate cells are liver cells, activation, and then acute phase response signaling. You can see all of these are enriched in the population with Down syndrome. And here's just one example, this atom TSL2. But it can be difficult at times to know how to interpret some of these pathways when we're looking in the plasma, right? So a lot of the gene expression enrichment tools are based on cells or tissues. They're based on RNA. They're not really based, for the most part, on proteins being secreted into the plasma. So how do we interpret some of this? It can be difficult, and we set out to find maybe some other studies that were looking at biosignatures of liver dysfunction. And then last spring, this paper was published looking at proteomic signatures of liver dysfunction in a pretty large cohort of individuals using the same SomaScan platform for proteomics that we use. And so we can really do kind of a one-to-one comparison and say, okay, which biosignatures that they see in their population are we also seeing in ours? And they generated a fibrosis signature with 100 features or so, and then a steatosis signature with fewer features in it. And we took those and used those to perform gene set enrichment analysis on the plasma proteome of individuals with Down syndrome. And what we found was significant enrichment for both of these signatures, right? Indicative of the idea that individuals with Down syndrome have plasma proteomic profiling that looks a lot like individuals that have bona fide liver disease that was confirmed via biopsies. And so just to show a couple of examples, again, IGF-1, which I already mentioned, this is one of their most downregulated in this fibrotic cohort. And here you can see the trends in the data from their group. And then this is what the Human Trisomy Project data look like, right? So down in the trisomy 21 relative to the controls, similar to what we see for this group with the fibrotic, right? Conversely, they see upregulation of a negative regulator of IGF-1, IGF-BP7, and again, we see the same elevation of this in the plasma of individuals with Down syndrome. I just wanted to dwell on this a little more. There's another group that has used, again, the same platform, the SomaScan platform, to actually stage liver disease using the IGF family members. So here you can see the stage of fibrosis and then increasing effects on the lower levels of IGF-1, and then a similar increase in the IGF-BP7 levels as we see progression of liver disease. And then just one more example here, this thrombospondin 2, that's really strongly elevated in this group, and we see the same elevation in people with Down syndrome. And I wanted to point this out because this is a marker. So here we're looking at control individuals, two different types of liver cancer that we can kind of ignore for the purposes of this talk, but then NASH, which is non-alcoholic steatohepatitis. And you can see here that this is elevated. And in fact, that signal alone actually has pretty good predictive value in this cohort for whether or not one has NASH, is the elevated levels of thrombospondin 2. So again, we think that we have a pretty strong set of these signatures, but we're going to need to do some more work to understand what the implications for those things are. So what do we know about liver function in Down syndrome? So mostly this has been studied through the prism of transient myeloproliferative disorder, or TMD, which is an alteration in blood cell metabolism. Early in life, they can be a precursor to some of those pediatric leukemias. There are cases of hepatitis, cholestasis, biliary atresia, vascular malformations. These are all super rare, and this is not what we're talking about. What we're talking about here is both in a pediatric and an adult population now, signs of widespread, albeit not progressive, alterations in liver function or liver signaling. So, has anyone else seen this? The answer is actually yes. There is a paper from 2017 from a group in Italy that found a pretty striking enrichment of non-alcoholic fatty liver disease, or NAFLD, in individuals with Down syndrome. So what they found was in their pediatric cohort, 45% of non-obese individuals with Down syndrome, kiddos, had NAFLD measured by ultrasound, and 82% of overweight and obese kids with Down syndrome had the same. Compared to a normal cohort, so again, this is not exactly the same study, but these are just standard values in the European cohort of 5.7 and 33%. So you can see in the non-obese here, we're looking at an 8 to 9-fold increase in the rates of NAFLD from this study. So again, it seems like there's some really promising signs, and so we decided to dig in and explore this further. So again, I mentioned this is non-progressive disease. We're not going to get liver biopsies on these kids who are otherwise healthy, and we frankly don't want those. And so we next set out to use human iPSCs. So these are great. We can take cells from a number of different sources and reprogram them kind of back in developmental time. We've done this with epithelial cells from the urine and also from blood cells. Introduce the Yamanaka factors, turn these back into iPSCs, and then we can drive them back out into lineages of our choice. And we're going to actually do that for hepatocytes. The good thing about these, they can provide an unlimited supply of material, be driven to these different lineages. They have a full copy of chromosome 21. That's going to be important when we get to the animal models. And we actually have one isogenic pair that came from an individual who's mosaic for Down syndrome. And so that's a pretty powerful tool there. And I just want to mention, we've heard this a couple times today, but we are developing with the DCC, the experimental models portal that will house data from things like iPSCs and mouse models, so that, at the end of the day, when all these data have been deposited, one can do an analysis like the one I'm going to walk you through here in the next 10 minutes or so. And all that will be available through these two related portals, and you'll be able to kind of get a sense across different tissue systems and across different models of Down syndrome, what kinds of signaling changes there are. Okay, so again, iPSCs can be driven to tons of different fates, including a number of different liver cells where we've already worked out conditions for this. I'm just going to talk very briefly about hepatocytes here today. And so we start from these iPSCs that are expressing, again, those Yamanaka factors. And then through a combination of chemicals and cytokines, chemokines, we can actually drive these throughout development, and we can get them into something that looks like mature hepatocytes. So they go through a definitive endoderm, through a hepatoblast stage, which is kind of a precursor to the hepatocytes, and then finally into these, what we're going to call IHEPs, induced hepatocytes. And we can track these by measuring the expression of different transcription factors that are present and driving these changes throughout time. And then ultimately we can measure kind of mature markers like albumin and cytochromes that should be expressed in these adult, more mature liver cells. So when we look here at D21, we're looking at these are euploid iPSCs versus T21 iPSCs. And again, we're just looking at albumin, one of these more mature markers that should be expressed in those hepatocytes, very low levels in the iPSCs, very low levels in the definitive endoderm, starting to come up, and this is a log scale, pretty substantially in the hepatoblast stage. And I think we're starting to see maybe a bit of a delta between the controls and the T21 cells. And then by the time we get to the hepatocytes, we have a pretty stark difference between the amount of albumin mRNA that's being made. This is qPCR in the controls versus the T21 hepatocytes. And the same is true for this CYP3A4, which is, again, just another marker of these. So we can make these hepatocyte-like cells. However, the induction of genes associated with the mature hepatocytes is lower. And in fact, if we go in now and use immunofluorescence to measure albumin, for example, we see good production of albumin in the D21 cells, much lower levels in those trisomic cells. And then conversely, if we do something like oil-O-red staining, which is in the orange here, to look for fat accumulation in these cells, we can see here pretty diffuse staining, maybe a couple of smaller lipid droplets in the control IHEPs, and then much more staining, much larger lipid droplets in the T21 IHEPs. And so again, we can use these iPSCs now to kind of identify where there may be some intrinsic deficits either in hepatocyte function or in the ability of cells to differentiate into those mature hepatocytes. Okay, so that's what we can do, an example of what we can do with iPSCs. And then we can also test this using animal models of Down syndrome. So I alluded to this earlier. This gets much more complicated. The genes on human chromosome 21 are spread across 3 different chromosomes in the mouse. They're on chromosome 10, chromosome 16, and chromosome 17. We are mostly focused in our group on DP16, which is about 120 protein coding genes that are orthologous to human chromosome 21, including our favorite interferon receptors. There are some segmental duplications and some freely segregating. All the ones that we're focused on today are these segmental duplications. But what's important here is this is an intact living organisms with interacting organ and tissue systems. And that for a lot of our studies, these mouse models have functional immune systems. So let's get right to it. If we do plasma metabolomics on these, we see, all these bile acids are elevated in the DP16 mice. And here's just one example in the cholic acid. If we look at some of those proteomic measurements, again, the ALT and AST, these liver enzymes, they're not elevated, but we do see that decrease in albumin, that hypoalbuminemia that we've seen, and that is characteristic of liver dysfunction. Okay, so again, the value of these animals is we can actually take their livers out and we can look at what's going on. And so when we take these animals out and we do H&E staining on them, what we see largely are these pathology that's associated with the portal triad. And I don't think there are probably too many histopathologists on here. I'm not one myself, but you can see here that there's more. This has got less purple kind of throughout. This has more purple. What this is a combination of arterial thickening, endothelial cell hyperplasia, biliary hyperplasia, and immune infiltrates. That's the bulk of the pathology that we see here. And you can see a pretty substantial increase in those features in the DP16 over the controls. So we can take those same livers, we can grind them up and ask what is the effect of those triplicated genes on gene expression. And what we see is it's pretty profound, about 1,500 DEGs in the DP16 livers with things going up and down. We do IPA, we find fibrosis signatures. This is another pathway analysis software, excuse me. Hepatic fibrosis in a couple of places. We see immune activation through the Th1, Th2. We see some metabolic pathways like GP6. So a lot of interesting things here that can allow us to ask further questions. So looking at the fibrosis now, we go back and we do a stain called picroserious red that binds to some of these collagen proteins, and then we image it with polarized light, and we see that there is in fact elevated fibrosis at baseline in these animals. And then we can actually integrate these data together. So now when we take the pathological score and ask, how does that affect the transcriptome? So now we're looking at basically the full change of the score relative to the transcript levels. We get some correlations here like FGF21 that we can see is elevated. So the more damage you have, the more FGF21. And this makes total sense because FGF21 is a protein that is elevated to play a protective role in damaged liver. So I just want to touch on one more thing that's hard to get away from in this group. We found that the DP16 mice had elevated interferon signaling via the transcriptome in their livers. And you can see here just a handful of these genes. So this is our kind of favorite hypothesis. There's these 4 interferon receptors on chromosome 21 and chromosome 16 in the mice. Those are triplicated. This leads to elevation of ISGs and ultimately driving pathology in various tissues. That's our hypothesis. We made a mouse that we published last year where we knocked out this whole cluster. And that allows us to create a cohort of animals where we have our DP16, we have our wild type, and then we have a DP16 2X IFNR. So we've just taken the copy number from 3 back to 2 in those animals. And we can ask what kinds of pathology is rescued. In that paper, we showed it rescues gene expression changes, a dysregulated antiviral response, CHDs, developmental delays, cognitive deficits, craniofacial patterning. So what happens to the liver function here? And given that strong interferon signature, we thought we might see a pretty solid rescue. That's not what we see. And so if we look at cell injury here, you can see here that it's elevated in both the DP16 and the norm, the 2X IFNR relative to the wild type. Same is true for the inflammatory response. If anything, there's a trend towards an increase. Same thing for these reactive changes. Again, these are some of those features that I described, and these are just more elevated. And that leads to this overall score where there is no rescue in these animals. Okay, so a quick summary here. The HTP data have revealed signs of altered hepatic function in Down syndrome through plasma metabolomics, particularly the bile acids, as well as proteomics. The T21-derived IHEPs produce lower levels of mature markers consistent with some intrinsic defect, and we're not sure what that is yet. And the DP16 mice have elevated plasma bile acid levels along with liver pathology, and we have much more data I didn't have time to show today. So the integration of data across these model systems can inform and accelerate research. And I'll just take 1 minute to do some acknowledgements. That was a lot of data that took a lot of people to generate. Lauren is a grad student in my lab who led all of the mouse work. Brian is a research associate at Cernic in Joaquin's lab who led the iPSC work. Again, a huge team. I don't have time to name everyone that helps with all the human, mouse, iPSC data generation, and then data analysis, and then all of our funders. And I'm happy to take questions. Thank you.
Thank you, Kelly, for a great presentation, showing how there's still room for major discoveries in the study of Down syndrome, including dysfunction of a key organ, right? With multiple potential impacts, such as that decreased IGF-1 levels that you show, which may be associated with maybe driving the stunted growth phenotype of Down syndrome. IGF-1, a major mitogen for human development. So we do have time for questions. We're doing well on time. We have another scientific presentation coming up. Any questions or comments for Dr. Kelly Sullivan?
Joaquin, can I ask a question?
Please, go ahead, John. How are you doing?
Good, how are you? I realize I'm taking advantage of the fact that I'm also on the panel. So Kelly, I think that's really fascinating data. Couple of things to ask you. Do you see any abnormality with bilirubin, either direct bilirubin or indirect bilirubin? And number two, you showed that there is a signature in terms of the proteomic profiling similar to NASH. So which of the genes that define that signature is actually on chromosome 21?
Great. So we have looked at bilirubin. There are some trends toward perturbations, but we don't see statistically significant changes either in the humans or the mice. And again, this is another one of these questions. I've talked to a couple of hepatologists, and we have some features that are consistent with canonical liver dysfunction and other ones that are different, right? And I don't think this is unique among different conditions in the Down syndrome population where we see certain things where maybe all the clinical hallmarks aren't present there. Which of the genes on chromosome 21? That is a great question. And again, back to my first slide with trisomy 21 in the black box. I have not found a slide on chromosome 21 that is a part of one of these signatures, right? And so I think most of what we're seeing there, one of those signatures was more NASH-like, one was more fibrotic-like. And what I think we're seeing there are, again, the signs of the disease. Those are not necessarily the causal proteins that we're seeing that are changed. We're seeing the consequences of those. And so I think this is actually a place where the mouse models have the potential to be really powerful, is we can measure these across a developmental trajectory and we can figure out what's changing before we get to full dysfunction, damage, where the proteomic signatures are starting to muddy the waters, and we'll never figure out which of those are cause and which are consequence.
Right.
So that's a great question, John, and we can already conclude that of the 200 plus protein coding genes on chromosome 21, it has to be one of the 120 or so triplicating the DP16 model, right? Because it has that liver fibrosis, liver pathology. And now out of those 120, we can subtract the 4 interferon receptors. It's not it. So we only have 116 genes to go. So it will be an interesting set of experiments to narrow down the genomic region farther. Yeah. Any other questions or comments for Dr. Kelly Sullivan before we transition to the last part of today's webinar?
Hi, Yokin. I have just one question. Shuja from India. That is,
Okay, I want to express my personal gratitude for you signing into this webinar at what seems to me like very late at night in Calcutta. What time is in Calcutta right now?
oh, it's almost 11 p.m.
There you go. Talk about a committed Down syndrome researcher. Go ahead, Sujay, with your question.
Thank you. My question is, age is an important variable of liver function determinants. So the result that Kelly exhibited is whether is age adjusted calculations or not, first. And my second question is there any differences in D21 and T21 liver metabolomic outcome through the ages? That means the difference calculated at the children and difference in adult is almost uniforms, or there is some variation?
Yeah, this is a great question. So your first question, it is age adjusted. So everything we did is age, sex, and BMI adjusted for the human data. As far as what those age trajectories are for the individual features, I would have to go and look very carefully at this. We've done some of these analyses for both the proteomic and the metabolomic data. Joaquin, you can comment on this maybe better than I can. My recollection is that a lot of the metabolomic stuff had less of an age component to it. And I don't remember the bile acids in particular having a strong age effect for those. But I would welcome your input on that, Joaquin. You're closer to that project than I am.
Well, as you explained, the data is age and sex adjusted. So even after age adjustment, the data is there. Now, the report from the Italian group that was very interesting, showing NAFLD, right? I believe that was mostly a pediatric population, Kelly, if I remember correctly. So I think, Sujay, this is happening early in life.
Yeah, the Italian paper is all pediatric.
Yeah. So it's happening early in life. So is it part of the precocious aging phenotype of Down syndrome? It may be, but it is happening very early in life. And somewhat related to what John was asking about bilirubin, there are a few reports, there are small sample sizes, about increased rates of jaundice in newborns with Down syndrome. Again, not perhaps to the sample sizes that we'd like to see, but I think the question is worth asking, John, whether, with large sample sizes like these cohorts that include Project is finding, if we were to do an analysis of bilirubin, and jaundice in those first few months of life, whether those trends will reproduce.
Yeah,
And
actually, the age-specific outcome will help the clinicians to design the management separately for the children and separate for the adult with Down syndrome.
yeah,
Correct.
and I would just
So go ahead, Kelly.
no, I just wanted to reiterate, you know, one of the interesting things here that we're seeing, this is not progressive, right? So
Okay.
so we're not seeing liver failure in the adult population with Down syndrome. And so again, what we're measuring here is something that's a little different than what we're seeing in the kind of Western adult population where, you know, NAFLD is progressing through fibrosis and ultimately into liver failure for some of those individuals. So I think whatever the age component is, it's present in both children and adults, and we have some questions about how that changes over time.
Yeah,
Thank you, Jay, for the question. Very important.
thank you. Thank you. It's a nice presentation.
Very good. If there are no other questions, then yeah, we'll move here to the last part of this webinar, which is another scientific presentation from another Include funded scientist who has data in the Data Hub and is doing great discoveries with those data sets. So this is Dr. John Yang. Are you, John, are you ready to share your screen? Very good. There we go. Great. And if you want to go to your first slide.
Can you hear me?
Yeah, we can hear you, but your slide is not sharing properly. Will you try again? We started there from, oh, there you go. Very good. So John comes to us from the Comprehensive Cancer Center at St. Jude's Children's Research Hospital, and he'll be presenting today on the genomic landscape of Down syndrome-associated acute lymphoblastic leukemia in children. So Jan, the podium is all yours.
Thank you, Joaquin. I hope you can see my full presentation mode.
Yeah, it is showing correctly. Thank you.
Great. Okay, so as Joaquin mentioned already, my lab is interested in pediatric cancer, pediatric leukemia in particular, and this aligns with our institution focus on various different cancers during childhood. So the work I'm going to present to you is a collaboration between my group and Karen Rabin at Baylor College of Medicine, and also Philip Lupo at the same institution. And the work cannot be done without the support from the Children's Oncology Group, which I'll talk about just in a minute. Here's the outline of what I want to discuss. I will give a very brief introduction about Down syndrome and its relationship to acute lymphoblastic leukemia in children. I'll talk about our work using genomics to define molecular subtypes of ALL kids with Down syndrome. I'll talk about the overall mutational landscape in DS-ALL. I'll also specifically talk about the molecular heterogeneity in Down syndrome ALL children carrying a specific translocation involving a gene on chromosome X called CRLF2. I'll talk about the genomic features of a Down syndrome ALL versus ALL children without Down syndrome. In the end, I will also mention a few analyses we did to identify prognostic factors associated with the Down syndrome ALL. And this is meant to be an interactive session, so feel free to interrupt me if you have any burning questions during my presentation, but certainly you can also save your question until the end. So the relationship between Down syndrome and acute lymphoblastic leukemia has been established for a very long time. It was first reported back in the 30s. The first systematic study was back in 1957, which showed that there is about 20-fold increased risk of developing leukemia during childhood, both acute lymphoblastic leukemia and also myeloid leukemia, AML. The cumulative risk is about 2% by the age of 5. So you can see the populational attributable risk is still relatively low in terms of developing ALL. However, this is much, much higher than the general population, meaning the kids without Down syndrome. And Down syndrome ALL comprises about 2% of pediatric ALL, about 10% of pediatric AML. So by all means, it's a relatively rare entity. However, the etiology of these two different types of leukemia are so unique, really begs the question, what are the biological features defining this unique pathogenesis compared to ALL or AML without Down syndrome background. So with the support of the Gabriela Miller and Kids First initiative from the NIH and also the INCLUDE initiative, and in collaboration with the Children's Oncology Group, we put together this very, very unique cohort of patients, about 300 kids with both Down syndrome and a diagnosis of acute lymphoblastic leukemia from various trials run by COG or the Children's Oncology Group. So every single patient was younger than 30 at the time of diagnosis of ALL. So we did whole transcriptome sequencing RNA-seq for 249 tumors. We also did whole genome sequencing for 275 tumors, 269 germline, of which 244 have both tumor and germline in a paired fashion. So this is a Venn diagram describing the overlap between different genotyping or genomic data sets, RNA-seq on the top, whole genome sequencing of the germline on the left, and whole genome sequencing of the tumor on the right. So as you can see, we have about close to 200 cases with all three types of data: RNA-seq of leukemia, germline whole genome sequencing, and also leukemia whole genome sequencing. So this is actually, I usually tell people something is one of the largest, but this is actually the largest genomic data sets for Down syndrome ALL, which, as you will see in the next few minutes, has really revealed some very interesting biology about this particular disease. So the first thing we want to do is to perform molecular subtype classification. Down syndrome ALL on the top, we have 4 different types of genomic features that we use to for subtype classification. The first is oncogene fusion. These are chromosomal translocations, which are detected by either RNA-seq as a fusion transcript or by paired whole genome sequencing through the analysis of a structural alteration. And the second type of analysis or feature is gene expression. So we use gene expression pattern to define certain molecular subtypes as well. This is purely based on RNA-seq. And then we also use what we call digital karyotyping, basically counting the number of chromosomes in the genome, and use this to define diploidy, whether it's a hyperdiploid or hypodiploid compared to the diploid genome. And in some cases, we do consider sequence mutation status to define subtype. As you can see, 8 subtypes were defined using oncogene fusion. 4 of them were defined by gene expression pattern. 1 is defined by karyotyping, namely high hyperdiploidy, and then 2 subtypes are defined by sequence mutations in 2 specific genes. So this is the summary of our subtype classification. I'm going to walk you through the plots because they are fairly convoluted. So the first thing that really caught our attention is the huge number of individuals with leukemia carrying specific rearrangement involving a gene CRLF2, and this is on chromosome X. As you can see, about more than 50% of the patients, or more than half of them, have specific rearrangement of this gene. And this is also showing on the oncoplot on the right. So on this oncoplot, each vertical column is a patient sample or a case, and each horizontal bar is a different molecular feature or genomic abnormality. So the first 2 rows are CRLF2 rearrangement. The very first one is rearrangement on chromosome X. So this is an intrachromosomal rearrangement between 2 adjacent genes, CRLF2 and P2RY8. And the second one is an interchromosomal rearrangement between chromosome 14 at IGH locus and CRLF2 on chromosome X. And the result of this, either one of these 2 rearrangement is this overexpression of CRLF2. So the CRLF2 expression is on the y-axis here. This this track highlighted by the red box. As you can see, every single case who had a CRF2 rearrangement had this massive upregulation of CRF2 expression compared to cases without CRF2 rearrangements. So another interesting group that we have identified is what we call a CEBP or unaltered. These are individuals, about 10% of Down syndrome LL cases. They have a series of genomic abnormality involving genes in the CEBP gene family. And we'll talk about that in more detail in a few minutes. So again, you can see on the uncle plot, there are a myriad of genomic abnormalities involving different members of the CEBP gene family. Many of them are CEBPD. Majority of them are actually CEBPD, either rearrangement of IGH or some other intrachromosomal abnormalities. And then we do have cases with rearrangement involving CEBPA or CEBPE. And the result of that is the overexpression of different members of the CEBP family. And here I'm specifically showing CEBPD overexpression. Cases with the rearrangements or enhancer hijacking events involving this gene had led to massive upregulation of its expression compared to the rest of cases. The last one I want to touch upon is IGF2BP1. So this is a gene, I believe, on chromosome 21. We have about 2% of, about 3% of the individuals with a very specific rearrangement between IGF2BP1 and IGH on chromosome 14. And the result of that is the overexpression of IGF2BP1, which is shown here, if you can see my cursor. What's also interesting is we have a large number of cases within the Down syndrome ALL cohorts. They do not have rearrangements of IGF2BP1, but instead they have a rearrangement involving two genes, ETV6 and RUNX1, which is on chromosome 21. And the ETV6 RUNX1 fusion, even though do not directly involve IGF2BP1 fusion, but this ETV6 RUNX1 fusion gene, for reasons we still do not understand, can lead to a massive upregulation of IGF2BP1 expression. So now again, overexpression of IGF2BP1 is a common feature in kids with Down syndrome ALL, both driven by IGF2BP1 translocation, as well as fusion gene between ETV6 and RUNX1. So now let's look at IGF2BP1 fusion specifically. So altogether, we have 7 cases with this fusion. As you can see, the rearrangement actually did not affect the coding region of IGF2BP1 gene at all. All rearrangements partner with IGH. The breakpoint at the IGH locus is actually the IGHJ gene segment. As I mentioned to you already, as a result of this rearrangements, you have this enhancer hijacking event connecting the constitutively active enhancer at the IGH locus, put that right next to the IGF2BP1 locus and driving its overexpression. And this is also seen in cases with ETV6 RUNX1 translocation. All right, what about CEBPD? I mentioned that earlier on the uncle plot. So altogether, I think we have about 11% of the individuals. So here I'm looking at the CEBPD rearrangement specifically, and you can see a majority of the cases rearranged to the IGH locus, again the J segment on chromosome 14. And most of the breakpoints are actually adjacent, are within the adjacent gene called spider. So this is a CEBPD gene, which is very, very small. And spider gene, which is upstream of CEBPD, is much bigger, but then your rearrangements is somewhat clustered within the first or the last few exons of spider 1 adjacent to CEBPD. The overexpression is expected. I'm showing here on the dot plot on the bottom right corner. what's interesting is when we perform the unsupervised hierarchical clustering analysis of gene expression of all the Down syndrome ALL cases, we observed that there are 8 cases that had a gene expression pattern that is very similar to the cases with CEBPD rearrangement. However, they actually do not have any CEBPD translocation. And what's really interesting is they also did not show high expression of a CEBPD. So looking into this group of 8 cases specifically, what we identified was that 6 of them have overexpression of CEBPA gene. 1 of them have overexpression of CEBPE, and there's 1 with the unknown overexpression, at least not within the members of a CEBP family. So if we focus on the CEBPA overexpressors, out of the 6 cases, 1 had a rearrangement with the IGH, again on chromosome 14, and then 3 of them actually had downstream small indel based on the somatic genomic analysis. And the CEBPE gene rearrangement involved T cell receptor A alpha. So now let's look at the CEBPA overexpression. Like I mentioned earlier, there are 6 cases showing overexpression of CEBPA, which are the purple dots. And these cases obviously don't have overexpression of other members of the CEBP family. So 4 of the 6 cases with the CEBPA deregulation had IGH rearrangement. And then you actually have a few cases, especially these 2. 1 actually had an 8 base pair insertion, another had a 54 base pair insertion. But one thing that's striking to me is how close these 2 insertion events are to each other in 2 different cases, highlighting the possibility of a hotspot right here that is particularly susceptible to genomic abnormality with a result of enhancer hijacking and CEBPA overexpression in the context of ALL. This is a CEBPE. Like I mentioned earlier, CEBPE is a very small gene, and this is a rearrangement with the T cell receptor alpha gene, driving overexpression of CEBPE compared to cases without this specific type of genomic abnormality. So what about other concurrent genomic features in the group with the C alteration involving genes in the CEBP family, either CEBPA, CEBPE, or CEBPD? So one thing that really stood out to us is there is an overrepresentation of mutation involving the gene FLT3. So 42.3% of the CEBP altered cases have alteration involving this gene. And this is almost 10-fold higher compared to other subtypes of Down syndrome ALL. The alteration in the CEBP altered subtype are mostly in the JAK membrane domain or the tyrosine kinase domain of FLT3, pointing to potential functional consequences of this mutation. What's really interesting is that even for the cases that did not have a somatic mutation in in FLT3, they still had an overexpression of the wild type FLT3, again pointing to the possibility of activation of the FLT3 signaling in this particular subtype of Down syndrome ALL. So this is a mutation landscape of DSLL, about 200 cases. It goes without saying this is a very expensive slide with all the whole genome sequencing that's performed. So it's also a very busy slide with a lot of data. So I don't want to go into every single gene, but instead I'm gonna spend a little bit of time highlighting a couple of key findings. One thing is the JAK2 mutation seems to be very high frequency in Down syndrome ALL. And in particular, the JAK2 mutation is exclusively seen in cases with CRLF2 rearrangement, I think there's definitely interaction between JAK2 mutation or JAK2 signaling and CRF2 signaling. We also see some CRF2 mutations and also mutation in a transcription factor known as IKZF1. Again, they are more frequent in the CRF2 rearranged cases compared to Down syndrome ALL cases without abnormality involving the CRF2 gene. And I mentioned already the FLT3 mutation seems to be particularly enriched in cases with CEBP alteration. And in that particular subtype, we also saw a higher percentage of a number of genes involved in epigenetic regulation, SETD2 and KDM6A. So the CRF2 mutation, this is a mutation only seen in the CRF2 rearranged cases. It's about 17 out of the 19 cases are in that rearranged group. 15 of the 17 cases are is highly conserved. This F232C mutation is 15 out of the 17 cases, and it's always occurred on the rearranged allele. So therefore, this is actually not only mutated but also overexpressed. Again, that's very interesting in terms of the functional consequences of this specific mutation event. So now if you look at CRF2 rearranged cases, accounting for about 50% of Down syndrome ALL. And we did a t-SNP plot to look at the molecular heterogeneity within this group. You can see that they are very clearly separated from each other as two distinct clusters. The cluster on the top, well, actually, let's talk about the cluster at the bottom first. So these cases, they have CR2 rearrangement, but more importantly, they also have a gene expression pattern that is very similar to ALL cases with the BCR-ABL fusion gene or the Philadelphia positive ALL. So we call them BCR-ABL-like ALL. So these cases, CRF2 rearrangement and with the gene expression pattern similar to BCR-ABL. And then the rest of the cases, they have CRF2 rearrangement, but for reasons we still do not understand, they do not have the BCR-ABL gene expression signature. So within this group, I mentioned already, they have a lot of alteration in IKZF1, EBF1, BP1, USP9X, etc. And this is particularly true in the rearranged cases that harbor the BCR-ABL gene expression signature. So now, if we were to compare Down syndrome ALL versus non-Down syndrome ALL, remember at the very beginning we talked about the Down syndrome ALL have a very distinct molecular etiology compared to ALL cases without Down syndrome. So what we're hoping to do is to compare the genomic feature between the two groups, trying to provide some insights into the pathogenesis of ALL with the different origins. So one feature obviously is the CRF2 rearrangement, 54% versus let's see, maybe less than 10% in the non-Down syndrome ALL. So the pie chart on the right is non-Down syndrome ALL, and the pie chart on the left is Down syndrome ALL. So the frequency of CRF2 almost tenfold higher in cases with Down syndrome compared to ALL cases without Down syndrome. The CEBP altered subtype and IGH rearranged with IGF2 BP1, they're extremely overrepresented in Down syndrome ALL. Other subtypes of ALL, such as high hyperdiploid, the BCR-ABL-like, the BCR-ABL, so on and so forth, they are actually underrepresented in Down syndrome ALL compared to kids with ALL without Down syndrome. So what about age of diagnosis? It looks like the onset of a CRF2 rearrangement positive ALL is about 5 years earlier in Down syndrome kids than non-Down syndrome kids. Again, we don't understand the biological causes of this, but this is the observation that we thought was quite interesting. Seems like there might be a different path to ALL leukemogenesis, even with the same, even when they end up with the same genomic abnormality. Another thing is what I talked about, the CRF2 rearrangement can happen in two different ways: intrachromosomal on chromosome X with P2RYA versus interchromosomal with IGH. So the P2RYA rearrangement of CRF2, which is intrachromosomal, seems to be a lot more frequent in Down syndrome ALL compared to non-Down syndrome ALL. The p-value is highly significant when you compare this to pie charts. And it also seems like the P2RYA CRF2 rearranged cases are younger compared to cases that do not carry this specific genomic abnormality. Looking at mutations in kids with Down syndrome ALL versus ALL kids without Down syndrome comparison, we did this for 52 genes and loci. Those that are at least 3% frequency in either one of the two cohorts I just mentioned. So most of the differences in frequency are based on subtype. For example, if JAK2 is more frequent, that's probably because JAK2 mutation is overrepresented in CRF2 rearrangement, which is overrepresented in Down syndrome compared to non-Down syndrome. So the results align really well with the differences in molecular subtypes between Down syndrome kids with ALL versus ALL without Down syndrome. So what about prognostic impact of molecular subtypes of Down syndrome ALL? So as I mentioned, we have almost close to a dozen different subtypes. That's why this event-free survival curve or Kaplan-Meier curve looks so busy, because there are many different groups. There are clearly differences in terms of their event-free survival or overall survival. However, our sample size is still relatively small, so it's hard to make a definitive conclusions in terms of which group should be upgraded to a higher risk of therapy compared to intermediate or low risk of therapy. But one thing that's quite striking was the difference between the two clusters within CRF2 rearranged. As I mentioned to you already, we can subdivide CRF2 rearranged cases into either carrying the BCR-ABL gene expression signature versus not BCR-ABL gene expression signature. So CRF2 rearranged cases, if they carry a gene expression signature that is similar to Philadelphia chromosome positive leukemia, their prognosis is very, very bad compared to the blue curve, which are CRF2 rearranged cases without this gene expression signature. So we actually have been in discussion with the Children's Oncology Group, thinking about ways to incorporate this finding into the next generation of leukemia therapy for children with the Down syndrome, whether or not this should be a risk factor to be integrated in their stratification for differential therapy and treatment regimens. So to summarize what I talked about today, clearly there's a distinct spectrum of molecular subtypes within Down syndrome ALL. We have 15 subtypes identified. CRF2 rearrangement is the top hit, 50% of the cases. Rearrangement involving IGF2BP1 and CBP genes are highly enriched in Down syndrome ALL. The BCR-ABL like gene expression signature seems to predict outcome, particularly in kids with the CRF2 rearranged Down syndrome ALL. There are a number of different genes that are enriching that group as well. I didn't have time to talk about our analysis of non-disjunct error in trisomy 21 and how that's related to Down syndrome ALL genetic subtype, but hopefully will be another occasion to share the data with everybody. So this work involves a large number of individuals. We have about 300 patients and families. Obviously, the collaboration with Karen Rabin and Philip Lupo and Baylor and Texas Children, Emory, University of Pittsburgh, and many others listed here on this slide. And also want to thank the funding sources, particularly the INCLUDE Project and also Kids First through the Gabriela Miller initiative. So I'll probably stop here and be happy to take any questions.
Thank you, John. That was great. A clear example how genomics studies supported by the Kids First and the Include project can help us understand the theology of important co-occurring conditions here in Down syndrome. And I see that we have a hand up. This would be Dr. Matthew Galbraith. Matt, go ahead.
Hi, Jen. I think that was pretty interesting stuff. I'm trying to think about this and place it in the context of things like TMD. And we've published on a higher prevalence of clonal hematopoiesis in individuals with Down syndrome. And you know, in that context, we think about some of these clones that appear to be growing out as having a differential selection or survival in people with Down syndrome versus their ability to act as driver mutations or you know, actually driving it. And I think what you're seeing here is probably the net effect of both of those things, that they may be somewhat weaker drivers in the typical population, but they could be stronger drivers in the Down syndrome population, or it could simply be that there's less selection against them in that competition in the early hematopoiesis. And I'm just wondering whether you have any additional thoughts on that. And I think this, right, our reports of clonal hematopoiesis, we don't know that any of them went on to have TMD or leukemias, but I think this is really important to have some sort of long-term follow-up on some of these people, and whether you can see that clonal hematopoiesis of, say, the BCR-ABL-like phenotype, and whether you could do something about it earlier on. And I don't think anything, ours was a targeted panel, so I don't think any of those rearrangements and things are even being monitored in the context of things like clonal hematopoiesis or TMD.
Yeah, thanks, Matthew. That's a very important point. So a couple of things. One is CHIP, clonal hematopoiesis. So we, as you have seen already, we do not see any mutation in the typical CHIP genes, DM3A and TP53, for example. And usually, if I understand correctly, for AML, if they have those, you have the CHIP, and eventually they will have a somatic mutation in those genes in the bulk AML samples, and we don't see that in the AML context. I do agree with you. I think especially for the CRF2, I think the CRF2 rearrangements clearly happens first, and probably shaping the pathogenesis or the pathway towards leukemogenesis. And JAK2 probably comes in a little bit later after CRF2. And I think CRF2, the way I'm thinking about it, is probably the driver, and then on top of that you have JAK2, and then together that really drives the oncogenesis. And this is probably also true for the CEBP alteration and FLT3. I mean, CEBP is a transcription factor, very important for myeloid differentiation, especially CEBPD, CEBPE. So you have a somewhat skewed differentiation, and then now you have a FLT3, which probably tip you over the edge in terms of leukemogenesis, although we haven't done any experiments to model that.
Thank you, John. I see Adam has a question as well.
Yeah, great presentation and really synthesis across the projects. If I understood correctly, the CEBP or any of the CEBPs, you know, essentially have a differential sort of differentiating context, whether in the setting of T21 or disomy as a context. And I started wondering whether, you know, could it be the case that you could screen, you know, a participant with trisomy 21 to assess, you know, whether their trisomy 21 context is sort of more fertile ground for a leukemia initiation setting versus not, right? You know, so in advance of the driver mutation potentially setting, you know, are there sort of trisomy 21 features that are distinct across patients that might drive predisposition to the occurrence of the leukemia in that setting? Does that make sense, what I'm asking?
So are you asking whether or not they have a different patterns of
essentially, like, the question is like, why, why does it only occur in some Down syndrome patients, right? So essentially, are there features that could be detected in advance of a leukemia initiation context of increased risk of a trisomy participant, for example, in a study? That make sense?
Yes, it does. I think that's why we are so excited to dive into the INCLUDE data sets, right? Because there are thousands of individuals with Down syndrome, but without acute lymphoblastic leukemia.
Yeah.
Actually, even before that, I think comparing Down syndrome kids with ALL versus AML will be very interesting already. So
Right.
why some of them developed myeloid malignancy versus lymphoid malignancy,
Right.
but then to talk about individuals without malignancy at all will be very interesting. I have to think about exactly how.
It's almost like I was wondering if there's a differentiation potential setting
I mean,
that you could test, right, within the context of the.
They all have a growth, gross chromosomal gain on 21. So it's possible they have some focal events on chromosome 21 at the germline level. Maybe that will differentiate ALL risk versus other conditions. But like I said, I'll be very.
I think it's super exciting, and I was just sort of connecting to the earlier presentation where it's clear, right, that there isn't just this linear process of you have an extra chromosome, so you have extra genes expressed, right? And so that, it just really sparked my imagination of, could there be a sort of biomarker of differentiation potential or not within the context of the blood that compartment that might be a feature of the trisomy 21 output that sets the stage for a leukemic hit, to be selected for. So I think it's a really exciting space.
Thank you for the strong interaction. And we have reached the top of the hour, and we're mindful that people need to get back to work and do meaningful work. So I want to thank to all the presenters today, to Hui-Ching, Adam, Kelly, and John for contributing to today's webinar. And yeah, please stay tuned. And we'll have another webinar, of course, in the fall. We're on a spring fall. We're trying to sync this with the equinox, these webinars. So we look forward to reconnecting with everybody, in fall later this year. Thanks, everybody. Have a great time.